chromosomes, and our linked genes are on the same chromosomes. So the example that we're going to look at today are obviously our independent genes, the ones that are found on different chromosomes. Okay, so independent of each other, they are not found on the same chromosome. So let's take a look at this example on the screen here. Here we have two different genetic traits in peas. Okay, so we're not looking at humans, we're looking at peas today. So the two genetic traits that are independent of each other in peas is the pea color, which is found on this set of homologous chromosomes in the pea, and pea texture, which is found in this set of homologous chromosomes in the pea. Okay, so each of these genes, the pea color gene and the pea texture gene, are found on different homologous chromosomes. And you'll also notice that for each of those genes, they each have a dominant and a recessive allele for that trait. So if we're looking at pea color, looking at the different allele allocations here, I can see that the yellow color is dominant, whereas the green is recessive, represented by the capital Y and the lowercase y. If I look over at pea texture, I can see that the allele representations here are showing me that smooth peas, represented by the capital R, are dominant, and wrinkled peas are recessive, represented by the lowercase r. So we're going to use these alleles and work through an example of dihybrid inheritance. Okay, so we're going to perform a dihybrid cross where we will cross a homozygous dominant for both traits with a homozygous recessive for both traits. So just think about what we already know about homozygous dominant and homozygous recessive. That means that for both of these traits that we're looking at, pea color and pea texture, if they are homozygous, they must have two copies of the same allele for each trait. If we're looking at the homozygous dominant, this particular pea is going to have the phenotype of yellow and, if my memory serves me correct, smooth peas. Okay, we do. All right, so yellow and smooth. They were the two dominant traits, and because they are homozygous dominant, their genotype, to get the yellow color, they must have two capital Y's, and to get the smooth phenotype, they must have the two capital R's. If we're looking at the homozygous recessive pea, the homozygous recessive traits were green for color and wrinkled for texture. And again, because they are homozygous recessive, they need to have two copies of the lowercase y to have the green phenotype, and they need to have two copies of the lowercase r to have the wrinkled phenotype. If we then cross those individuals, so thinking about what alleles they can pass on to future generations, we can see that our yellow smooth pea, the only possibility that it can pass on to its future offspring is a capital Y for the pea color and a capital R for the pea texture. So this pea, the yellow smooth pea, can only pass on a capital Y and a capital R. Our green wrinkled pea, on the other hand, can only pass on the lowercase y and the lowercase r. Which gives us an F1 generation of all offspring having capital Y, lowercase y, capital R, lowercase r. So all of their offspring are going to be heterozygous for both traits. And because they're heterozygous, they're going to show the dominant phenotype for both traits, which would mean they would have yellow color, smooth texture. So crossing two homozygous individuals is pretty straightforward. But when it comes to crossing heterozygous individuals in a dihybrid cross, that's when things can get a little bit fiddly. So we're going to have a look at an example where we're crossing a heterozygous for both traits with another heterozygous for both traits. So both individuals have the same genotype and phenotype. So they're both yellow peas with a smooth texture. And because both of them are heterozygous, they have the same genotype of big Y, big R, little r. So let's have a look at what they could possibly pass on to future offspring. Remember, both of them have the same genotype, so both of them can pass on the same combinations. So our first combination is capital Y, capital R. So we've got a capital Y, capital R. Yep. Second combination could be little y, capital R. Third combination might be capital Y, little r. And our fourth combination is they could pass on both of those recessive alleles, the little y, little r. Now remember, both of these parents have the same genotype, so whatever this parent could pass on, this parent also has the same possibility of passing on the exact same thing. So let's have a look at what this might look like if we're drawing up a Punnett square so that we could potentially predict our phenotypic ratios for these offspring. So this is what a dihybrid cross Punnett square looks like. It is much bigger than what we would see with our regular monohybrid inheritance. But we still use the same rules. So you'll see that we are filling in the possible combinations in gametes for parent 1 along the top row, and then the parent gamete combinations for parent 2 down the side here. Okay, so those rules still apply. And then we still fill in a Punnett square exactly the same way as what we have been doing in the past. So we combine those two combinations from each parent to get a possible genotype for that offspring, and filling out the Punnett square as we go. Okay, so these will be the possible combinations that you can get by filling in the Punnett square. Now that just looks like a bunch of Y's and R's to me. So let's have a closer look at what these actually represent. Remember any square that contains the capital Y was yellow, and the capital R was smooth. And we also had the lowercase y, which was green, and the lowercase r, which was wrinkled. Okay, now obviously if we want to show a green phenotype, because it's recessive, we need 2 copies of the lowercase y, and the same with the wrinkled phenotype. If we want to show that, we need 2 copies of the lowercase r. So you're essentially looking through these boxes and trying to identify which peas will be yellow, which will be green, which will be smooth, and which will be wrinkled. That might help you out a little bit. Okay, so we can now clearly see which ones are yellow, which ones are smooth, which ones are green, which ones are wrinkled. So if we were to go through and count all of the all of the peas that show the different phenotypes, we're actually going to find that there are 4 combinations in total. Those 4 combinations in total are going to be used to represent our phenotypic ratio for this cross. So the 4 combinations that we can have is we can have a yellow smooth pea plant, we can have a yellow wrinkled pea plant, a green smooth pea plant, or a green wrinkled pea plant. So if we go through our Punnett square and count all of those up, I'm just going to swap through some colors here. So for yellow smooth, I'm going to put a little red line under it. So yellow and smooth, yellow and smooth, yellow and smooth, yellow and smooth. Okay, so that's 1, 2, 3, 4, 5, 6, 7, 8. Oh, I missed one. 9. So 9 yellow and smooth. Yellowed and wrinkled. We've got 1, 2, 3. Green and smooth. 1, 2, 3. And then green and wrinkled. Just one in the corner there down the bottom. Okay, so that actually leads us to having that 9 to 3 to 3 to 1 ratio. A little bit different to what we've seen in our monohybrid crosses before, but it means that if we are crossing two heterozygous individuals in a dihybrid cross, we can predict that the phenotypic ratios for their offspring are always going to be 9 to 3 to 3 to 1. There we go. So a phenotypic ratio of 9 to 3 to 3 to 1. As a general rule of thumb, when it comes to dihybrid inheritance and we're looking at independent genes, if you ever cross two heterozygous individuals together for both traits, the phenotypic ratio that we're going to see in their offspring is always going to be 9 to 3 to 3 to 1. Okay, so it's probably quite easy for you guys to remember that little fact there, this general rule. But it's not good enough to just remember that general rule. The other thing that you need to be able to identify, and this will come with practice, is what the 9 represents, what each of the 3 represents, and what the 1 in the ratio represents. Okay, so that's what's explained just below here. So the 9 in the ratio is representing that out of those 16 offspring in that massive dihybrid Punnett square that we just did, 9 of the 16 showed the dominant trait for both of those genes. So in the example we looked at, there were 9 pea plants that were yellow and smooth, which were both the dominant traits. In that Punnett square, 3 of those squares showed dominance for the first trait, but they were recessive for the second trait. So in the example we just did, there were 3 individuals or 3 peas that were yellow for the first trait, but were wrinkled, which was our second trait. The second 3 in the ratio represents that they were showing the recessive phenotype for the first trait and the dominant phenotype for the second trait. So in the example we just did, there were 3 peas that were green, the recessive for the first trait, but smooth, which was the dominant for the second trait. And the 1 in the ratio is representing that one square right down in the bottom right hand corner where you were looking at a pea that was recessive for both traits, so green and wrinkled. Okay, so 9 to 3 to 3 to 1. Don't just memorize the ratio, understand what each of those numbers means. So from this point on, I am going to walk you through another example of dihybrid inheritance. If you feel that you've got a good enough understanding at this point that you don't need to watch this second example, you are welcome to stop the video now and continue on with your own practice questions. If you do want to go through another example, please feel free to stay watching this set of notes. Thanks, guys.